All right, let's talk about one of the biggest but also most invisible health threats we're facing today. It's called antimicrobial resistance, or AMR for short. You've probably heard of them as superbugs. These are basically bacteria that are getting really good at outsmarting our best medicines. So in this explainer, we're going to break down just how serious this crisis is and then dive into the amazing science that's helping us fight back. Okay, to really get a sense of the scale here, let me start with just one number: 4.95 million. That is the estimated number of deaths that were linked to bacterial antimicrobial resistance. And get this, that's just in a single year, 2019. So this isn't some far-off, futuristic problem. No, it is happening right now, and it's threatening the very foundations of modern medicine. So what exactly is AMR? Well, to put it simply, it's when tiny little microbes, like bacteria, start evolving defenses against the very drugs we designed to kill them. They basically learn how to survive the attack. And when that happens, our treatments, like antibiotics, they just stop working. And that makes everything from a routine surgery to something as serious as chemotherapy way more dangerous. All right, so let's dig into how this actually happens. How does a simple little bacterium turn into one of these so-called superbugs? And believe me, the stakes here are incredibly high. This isn't just a problem you find in hospitals. The source material really drives us home. It's a threat to our health, sure, but also to our food security and even the environment. It's a pollution issue. So understanding how bacteria pull this off is absolutely critical, because the problem is just everywhere. Let's take a look at their bag of tricks. You see, bacteria are absolute masters of evolution. They've got three main ways they do this. First, you've got random mutations, just little errors when their DNA copies itself. Second, they can do something called recombination, where they swap sections of DNA. But the third one, this is the real game changer. It's called horizontal gene transfer. And it's wild. They can literally pass resistance genes to each other, almost like they're trading cards. And that is how resistance can just explode through a population so fast. And that idea of things spreading leads us to a super important concept. If you want to understand AMR, you can't just look at it through the lens of human medicine. You have to see it as what's called a One Health problem. And this picture right here, it shows you exactly what One Health means. See, these resistant bacteria, they don't care about boundaries. They can travel from a person in a hospital, into our wastewater, then out into rivers and soil, then into farm animals, and eventually right back to us on our plates or in our drinking water. It is one giant interconnected cycle. So you might be asking, what's making this whole thing worse? What's pouring fuel on the fire? Well, it's not just doctors over-prescribing antibiotics, though that's part of it. There's a whole bunch of things creating the perfect storm for resistance to grow. We're talking about things like poor sanitation, pollution from factories, even climate change, which is changing where diseases pop up. All of these pressures basically force bacteria to adapt and get stronger faster. Okay, so the problem is huge. It's complex, it's a little scary. But here's the good news. Our tools for fighting back are getting seriously sophisticated. So let's talk about our new secret weapon, the world of genomics. I mean, the leap in technology here is just massive. Think about it. The old way of doing things was so slow. You'd have to take a sample, grow the bacterium in a petri dish, and then test to see if an antibiotic killed it. Now, with genomic surveillance, it's a total revolution. We can just take a sample and almost instantly read the microbe's entire DNA. And that doesn't just tell us, yep, it's resistant. No, it tells us how it's resistant, where it came from, and even helps us predict where it might spread next. It's a game changer. And here's kind of what that whole process looks like. It all starts with collecting samples from people, from animals, from the environment. Then you sequence all the DNA in those samples. And this is the cool part. Powerful computer analysis, which is a field called bioinformatics, sifts through all that raw genetic code and turns it into information we can actually use. Actionable intelligence. This is how public health officials can spot an outbreak early, trace it back to the source, and figure out the best way to shut it down. Now, one of the most powerful tools in this new toolbox has a cool name, metagenomics, and it's exactly what it sounds like. Just imagine scooping up a bit of wastewater. With metagenomics, you can sequence the DNA of every single microbe in that sample, even the weird ones we can't grow in a lab. It gives us this incredible, big picture view of all the resistance genes that are just floating around out there in the environment. Okay, but with all this complexity, all these genes, all these bacteria, you can't possibly track everything, everywhere, all at once. It's just impossible. So to get around that, scientists have come up with a really clever strategy. So that really begs the question, right? If these resistance genes are spreading through countless different bacteria across all these different environments, How on earth do you track a problem that massive? How do you find the important signal in all of that noise? Well, the answer is actually pretty smart. Instead of trying to watch everything, you focus on one single indicator organism that can tell you a lot about what's going on. And in the global fight against AMR, that indicator is a bacterium you've definitely heard of, Escherichia coli, better known as E. coli.
So, why E. coli? What makes it so special? Well, it's almost like it was perfectly designed for this job. For starters, it's a natural resident of our guts and animal guts, too. It can be monitored in people who are sick and people who are healthy. It's tough. It survives in soil, water, you name it. It also causes a lot of common infections like UTIs. And here's the kicker. It's a major hub for acquiring and sharing those resistance genes we talked about. So by tracking just E. coli, we get a surprisingly accurate snapshot of the whole AMR situation. And this is where it gets really interesting. There's a debate going on. The source material points out that by focusing so much on just one type of resistant bacteria, even a good indicator like E. coli, we might be getting a slightly skewed picture. Are we accidentally creating blind spots? Could we be missing other dangerous bugs or new ways that resistance is spreading? It's a really important question that scientists are grappling with right now.
And all of this brings us to the final piece of the puzzle. We have the amazing science, we have the powerful technology, but actually putting it all into practice across the entire globe, well, that comes with a whole new set of challenges. Yeah, the biggest hurdles we face now aren't really scientific anymore. They're more about logistics and money. We're talking about not enough funding, a shortage of people who are actually trained in genomics, and a lack of the basic lab equipment and computers. And this is especially true in low and middle income countries, the very places often hit hardest by AMR. They just don't have the resources to set up this kind of high-tech surveillance. But it's not hopeless. There is a clear path forward. The strategy really rests on 4 key pillars. First, we need to integrate our surveillance, get the human, animal, and environmental data all talking to each other. Second, awareness and education. We have to teach everyone, from doctors to farmers, how to use antibiotics responsibly. Third, enhance capacity. We need to invest in labs and training, especially in those countries that need it most. And finally, number 4, strengthen stewardship by putting smart regulations in place to protect the antibiotics we still have. So, this is where we leave it. We have the genomic tools to literally read the enemy's playbook. We can track its movements, and we can even predict its next move. The technology isn't science fiction. It's here, today. So the final, and honestly the most important question is this. Do we as a global community have the will? Can we find the coordination and the investment needed to actually use this knowledge before it's too late?